Today I'm discussing expanding approaches across gastrointestinal cancers, as well as hematologic malignancies, with exciting developments that have furthered our understanding of these challenging diseases. First, the recent American Society for Clinical Oncology, or ASCO GI Cancer Symposium, was held in San Francisco, California, and important updates about GI cancers were developed and presented, and it should be acknowledged this is quite a heterogeneous group of cancers where novel therapies and combinations for a generally immunotherapy resistant set of cancers are now seeing some progress. So upper GI cancer types like esophageal and gastric cancer, as well as some mismatch repair deficient, so-called microsatellite unstable high, MSI-high colorectal cancers have been the relatively unusual subset that are immune responsive. We've seen the benefit in gastric and this MSI-high colorectal. This was a treatment with immune checkpoint alone or in combinations. They may see dramatic benefit, but others such as hepatocellular or BRAF mutated colorectal cancers may need to have other treatments besides immunotherapy. And these would include the multi-kinase targeted inhibitors, the small molecule drugs. So at the ASCO GI, we saw exciting data and analyses to suggest that early or long-term results in recurrent and metastatic diseases may be seen by layering PD-1 and CTLA-4 inhibitors onto the existing systemic agents, such as the EGFR inhibitor cetuximab or Erbitux. Given that our understanding of the biology of these subsets has improved, but is not yet universally settled, the field is challenged to continue segregating subsets by genomic or targeted dependencies, which can then be paired with particular treatments. Indeed, new scientific advances like these multi-omics strategies should enable further insights into risk stratification and identifying different subsets with appropriate tumor specimens in clinical trial designs to continue advancing this field. Now, looking ahead to coverage in the next issue, the European Hematology Association and European Bone Marrow Transplant 7th Chimeric Antigen Receptor or CAR T-cell meeting is going to provide the frontier for gene-modified cellular therapies. The agenda is packed with exciting new information extending beyond hematologic malignancies, where we have 5 FDA approvals currently of CAR T-cells in different types of blood cancers. And this rapidly growing experience is now being transferred into solid tumors. With the recent approval of tumor-infiltrating lymphocyte therapy, or TIL therapy for melanoma, we now see an explosion of possibilities, such as gene-modified autologous cell strategies. These include CAR T-cells for solid tumors, where we identify a particular receptor that may be useful in gliomas or breast cancer or pancreatic cancer. And this creates an innovative strategy to expand the cellular substrates for longevity and tumor-specific targeting, because these are truly living therapies. At present, CAR T-cells are a laborious and cumbersome exercise requiring GMP and FDA-cleared facilities, developing vectors, and particular chimeric antigen receptor treatments that target the tumor selectively, with the hope that they have limited toxicity on the normal tissue interface, which is where we might see some off-target effects. So as we gain greater experience with the affinity of CAR T-cells for the tumor target itself, as well as the expression profile in the various solid tumors, these innovative strategies need to be applied in patients with very careful monitoring. Given that hematologic cancers have had such a fantastic success with CAR T-cell strategies, we would expect further cellular approaches in clinical trials to extend these endpoints rather than simply have first-in-human or toxicity questions, and actually figure out whether this will work as an effective therapy for solid tumors. But this would be quite exciting due to the durability of these cellular approaches. And unlike long-term treatments for a year or more with the immune checkpoints, cellular approaches may only require one transfer of cell therapy and allow the cells to persist if they're produced the right way. So it's quite exciting to see these standard of care regimens extended, and CAR T cell field moving into solid tumors, and it's quite a welcome development to see this progression in the field.